Great. Okay, well, I'll get started then. So welcome everyone to the second Cognition and Natural Sensory Processing Workshop, CNSP 2022. Great to have some of you back and good to see some new people here. So before we kick off, I am just going to do a little bit of housekeeping. So briefly, I'm just going to introduce ourselves. So for those of you who attended last year, you might recognize a few faces here. Myself, Aaron, Giovanni and Nate organized the workshop last year. Luckily this year we have had Georgia and Stephanie to help us out, which has been great. So just a little breakdown of who you guys are. So we can see that the majority of people attending this year are either PhDs or postdocs. We've got quite a few master's students and undergrads as well as research assistants and some faculty. Just over 60% of people are going to attend the tutorials, which is great. So we're looking forward to them. And then what languages does everybody speak? So it's very clear that most people are doing their analysis these days using MATLAB or Python or R. There's a few people using JavaScript and some other languages, but the majority are through MATLAB, Python and R. And luckily the hands-on tutorials that will be given this year are again through MATLAB and Python. So hopefully that'll meet the needs of most of our attendees. And for those of you who aren't familiar with those languages, hopefully it'll give you an opportunity to pick up some new programming skills. So just a few points on how to interact during the workshop. So during the talks and webinars, we'd like you to use the Zoom Q&A function. So if you could just type in your questions during the talk into the Q&A window, and we'll get around to them then at the end of the talk. We can pose your questions then to the speaker, or if you'd like, you can raise your hand and ask the speaker directly yourself, and we'll try to accommodate everyone as best we can. During the tutorials, we'll take a more interactive approach. So this will just be like a normal Zoom meeting. We'll expect you to actually run the code that we're demonstrating in real time on your own machine. So please make sure that you've got all of the resources that you'll need for these tutorials if you haven't already done so. And Giovanni will go through them later, everything you need after the keynote talk. Then in terms of just kind of more casual communication during the workshop, we're going to use Slack again. This will give you an opportunity to ask questions and interact with other attendees and organizers. If you have any particularly overarching questions that you think would be interesting for the discussion on day 3, you can post them into the questions for day 3 channel. So the link for the Slack group is contained in an email titled Slack group during the CNSP workshop. I also have a link to it down here, which I can just put into the chat here. So, okay. So then on day 3, we'll have a discussion which will be organized in 2 parts. One focusing on open science and standardization, and the second part will look at current challenges with TRF analyses, and we'll conclude then with the Q&A session based on any of the questions that attendees have posted in the Slack. So as always, we're happy to get your feedback on how the workshop functions. We'll send around a questionnaire at the end so that you can help us make it better for future workshops. So just a brief overview then of the workshop itself. So today we're going to kick off with session 1 on investigating auditory processing with natural sound listening paradigms. And we'll have our keynote speaker, Dr. Liberty Hamilton. Then in the, well, I won't say afternoon because I don't know what time zone everyone's coming from, but at 6, Irish time, then we'll get into our hands-on tutorials on encoding and decoding models for neural signal analysis in MATLAB. So this is a multi-track tutorial. So basically, for those of you who attended last year, we'd encourage you to attend the intermediate workshop. And for those of you who are new to this, you should attend the beginner workshop. Tomorrow then, we'll have session 3 and 4. We'll focus on novel directions on models for neural signal analysis in natural sensory scenarios. And we'll hear from our young keynote speaker, Narayan Sankaran. We'll also have 2 short talks then from 2 other young investigators, Michael Thornton and Joshua Kulasingham. Then in session 4, the 2 latter speakers will give hands-on tutorials for encoding and decoding models in Python. So again, this will be a multi-track tutorial on deep neural networks and encoding and decoding models in eel-brain. And then on day 3, we'll have session 5, which will focus on current challenges, best practices, and future directions. And we have an invited speaker, Giacomo Berzo, and he'll talk about everything to do with open science and data standardization. Then we'll go into our 2-part discussion and the Q&A after that. So just to talk a little bit about the goals of the workshop. First and foremost, we set this up just to help students and researchers who are new to this, just to be able to learn in a hands-on way through didactic tutorials. We also see this as a way for researchers to connect with each other, particularly, during COVID, it's harder to travel, and I'm sure most of you aren't doing as much anymore. So it's a great opportunity to be able to interact in a more casual way through the Slack and through the talks and questions. And then we also see the workshop and the website that's come out of this as a kind of a resource for sharing data, code, methods, standardization techniques, and literature. So on that last point, you can find all of the resources to do with the workshop and topics around the workshop on the website under the resources tab. So Giovanni will go through this in a bit more detail after the keynote talk when he's explaining how to set up your environment. And then finally, just to give a bit of context into the motivation behind the workshop. So essentially, a lot of us have started using different types of analytical techniques to allow us to conduct neuroscientific experiments in more naturalistic environments. And really, we just see this workshop as a way of kind of bringing together a lot of the resources that we've talked about, and also provide a little bit of, training and tutorials for younger researchers and researchers that are new to the field in order to be able to access this type of research and conduct research in more naturalistic ways. And just to mention this lovely figure here was put together by one of our organizers, Erin, as part of a paper that we wrote last year after last year's workshop. And that paper is actually quite relevant to the workshop because it goes through a lot of the different considerations and techniques used in conducting naturalistic sensory experiments. So I'd recommend to have a look at that if any of you haven't seen it, as it's quite helpful. And actually a lot of the content in that paper came out of last year's workshop, just from some of the discussions we had, some of the questions we got in Slack and so on. So it might be quite useful for a lot of you. So without further ado, I'm going to introduce our keynote speaker to you now. So it's my pleasure to have with us Dr. Liberty Hamilton. Liberty is an assistant professor at the University of Texas at Austin. She has a joint appointment at the Department of Speech, Language and Hearing Science and the Department of Neurology. She holds an adjunct position in the Department of Neurosurgery at Baylor College of Medicine in Houston, Texas. Liberty obtained her PhD in neuroscience at the University of California, Berkeley in 2013 in Dr. Sharon Bao's lab, where she studied sound representation and plasticity in the auditory cortex using multi-unit electrophysiological recordings. She completed her postdoctoral training with Dr. Eddie Chang at the University of California, San Francisco, where she investigated how human auditory cortex represents speech sounds using intracranial recordings in adult patients with intractable epilepsy. Liberty's lab now studies how sounds are represented in the auditory cortex during natural speech perception and production, and how these representations change as a function of development, attention, and multimodal interactions with other sensory modalities. Her group uses a combination of intracranial recordings in children and adult patients with epilepsy, scalp EEG, and computational modeling to understand these processes. Liberty's lab has received funding from the NIH National Institute of Deafness and Other Communication Disorders, the Texas Speech Language Hearing Association, and the Coleman Flung Foundation. So today, Liberty will give us an overview of how linear encoding models such as TRFs can be used to understand natural speech and sound processing in the brain in multiple recording modalities. So with that, I'll hand it over to Liberty.
Great. Can you see me? Can you hear me?
Yes.
Okay, excellent. All right, thank you so much for being here, everyone. I'm so happy to give a talk here at the CNSP workshop. I think there's a lot of really interesting work going on here, and I hope that you see how powerful these methods can be for doing a lot of types of analyses that I think the folks in our audience are going to be sort of excited about. So let me just get started. And then if you'll let me know. Okay, how do I look in terms of slides?
Looks great.
Okay, great. So yes, thank you again for having me. I'm so excited to be here. And so as Mick just said, I have spent a lot of time in my career looking at how the brain processes natural sounds. And I've been doing this for a little while, both in animal and human models. And today I'm going to give you a little bit of an overview of what I have been doing in my lab, as well as in my postdoc. But the main idea that I'm interested in is how is it that we are able to take in ongoing sounds and turn them into meaningful language and communication. And so this is something that is happening right now. Hopefully you're taking in the sounds that I am saying into my microphone, and you are transforming those into a series of phonemes, words, syllables, etc., and hopefully getting something out of it, and then interpreting this and changing your behaviors, whether that's learning a new analysis or learning something new about the brain. So again, this is a complicated process. We start with an acoustic vibration at the level of the ear. So that might look something like this. This is an auditory waveform. So if you were to record your own voice in a program like Audacity, something like that, then you could see this waveform. And when the waveform goes up, that generally shows that you have sort of louder, more power in the sounds. And if it's totally flat here, that means it's silent. But this isn't really how our brain represents the sound, as far as we can tell. So what we think happens is that there are a series of steps where these acoustic vibrations get transformed into a spectrotemporal frequency decomposition. So for example, we can take this sound and then figure out what are the different frequencies that are present from low to high. And you can see this is the sentence, "Have you got enough blankets?" And so at some level, that spectrotemporal frequency decomposition gets turned into some what we call more invariant representation of phonemes. So I could say, "Have you got enough blankets?" Mick could say, "Have you got enough blankets?" We have very different voices. So we might have different pitch of our voice. We also have different accents, but you know that we're saying the same phonemes. And so you can interpret that as the same sentence. And so that's going on somewhere in the brain. And this is something that we'd like to figure out, sort of how that process works. So then, of course, you can keep going up this hierarchy to the syllable level, word, sentence, or even semantic level. And this involves various regions of the auditory pathway. And here, starting at the cochlea, going up through the brainstem, thalamus, and auditory cortex. One of the things that I want to give you a little bit of intuition for, though, before we get started on these MTRF models and speech tracking and encoding and decoding models, is I want to give you some kind of idea of how the brain actually is able to take in sounds and turn them into a frequency decomposition. So let me just switch over here, and I'm going to show you something a little bit different. So can you still see my desktop? Looks like yes. Okay, so here what I'm going to do, and hopefully you will be able to see this, is I am going to speak into my microphone. And this is a demo that shows you, as you continuously speak, what the different frequencies are that are present in the speech sounds that I'm saying. So hopefully the Zoom lag is not too bad, and you can see that as I speak, we have high frequency up at the top, lower frequency sounds at the bottom. And with speech, it's very complex. You can, maybe if you're very used to looking at these, you could tell which sounds I'm saying just based on this picture. But overall, this can be tricky, and this is something that you have to get used to. So let me show you a few things. If I say sounds that have high frequency content, like S, you just see this energy at the top of the plot. And so those are high frequency sounds. I could do the same for as in father, right? And that looks a little bit different. Now, if I said the vowel ah, e, oo, you can see my voice is warbling a little bit, but you can tell that that looks quite different, and it has these stacked frequencies that are characteristic of what vowels look like. This is going to be important if you read any papers that are looking at spectrotemporal receptive fields, because they're going to take advantage of this type of visualization. And I think this is really nice because it gives you a much more intuitive look at what it means to have a spectrogram. And I'll also paste this into the chat. I don't know if we can share with everyone. So there we go. So you can try this out on your own. I think it works well in Chrome and on some tablets. Doesn't work well on a phone. So don't try it on a phone, but on a normal browser, it should work pretty well. They also have things like you can click on each of these sounds and see how the flute looks. So there you see how as the flute goes up, you see the harmonics. So things like that. And then just as one more, in case you're still a little confused, if I whistled, and let's see if I can do this. So you see how that is more of a pure tone. So that is just a frequency moving up and down in that space. And so I like this as a demo because it kind of helps you get a feel for what's going on when you're looking at this spectrotemporal frequency decomposition. So if you see high frequency stuff like shh, that means that maybe that neuron or that brain area is responding to a sh sound. Whereas if it looks more like an ah, then maybe it's responding to lower vowels, things like that. Okay, so let's keep going. So here, hopefully you saw that one of the kind of aspects of this visualization and of natural speech in general is that it is continuous. So these, and in fact, if you talk to people who study linguistics, in natural speech, we have a lot of connected sounds. We don't necessarily have a space or a silence in between every single word that we say. They're actually quite continuous. They're also non-random. And in fact, if I want to go further than that, I would say that these sounds are very structured. So there are non-random elements of the way that the sound signal evolves over time. If it were random, it would sound more like white noise, just like a, that sort of, with no structure in it. And in fact, a lot of folks design stimuli that are random to kind of get rid of some of this, because this can be difficult to deal with statistically. And the final thing that's sometimes difficult to deal with is that speech and other natural sounds are often what's called autocorrelated. And what I mean by that is if you're looking at, so this is time on the x-axis, which is not really shown as an axis. But over time, you have this signal that's changing, but it doesn't change randomly. You know, you can look at this time point right here, and if you look at what came right before it, it's quite similar. So they're correlated with what was happening in the past. Of course, this happens at different timescales, but this is something that makes these type of stimuli difficult to analyze. And I think that has been why historically there was much more of a push for controlled stimuli that were not related to each other in any way. And in fact, you would get rid of a lot of this structure in order to not have to deal with it. But today I'm going to show you how we do use these multivariate temporal receptive field models, or STRF models, or encoding models to cope with this, and in fact to leverage the power of being able to look at a more naturalistic stimulus. So again, like I said, if you were doing a typical neuroscience experiment, not in this kind of continuous naturalistic framework, you might choose to play stimuli to someone that are pretty simple. So maybe you have someone listening, you're recording brain activity somehow, and you have them listen to syllables like ba, da, ga, and they're separated and we have space in between them and we manipulate them in specific ways so that we can do a contrast analysis. If we don't want to use phonemes, there are also folks who use these types of ripple sounds. So this is what the spectrogram of some of these kind of strange sounds look like. And these are used both in the human and the animal literature to try and get auditory receptive fields by just playing lots of different combinations of sounds that either have high frequency content or temporal content. So they're either changing a lot in the pitch or the time domain. And these can be efficient, but they're not like real speech. They're not like the sounds that we really encounter in the real world, unless you're, I guess, playing a lot of video games and just hearing lots of weird sounds all the time. So let's say you want to add some color to your world. And maybe you're more interested in what happens when people are listening to music. This is something with a lot of structure. Maybe you're interested in natural speech. So what happens when people are listening to podcasts? What's going on in their brain? What types of things is the brain paying attention to? Or maybe you're interested in natural sounds. So this could be people listening to birdsong. It could also be birds listening to birdsong. So this method, I hope you'll understand later, is very powerful and has actually been used in many, many different fields. And so the answer to, if we did want to look at these types of natural sounds, is instead of having to deal with sounds that are not correlated with one another, that are separated with lots of silence and maybe honestly quite boring to listen to, we can use encoding models. And one thing that I also want to point out is that these have a lot of different names. So you may see any one of these terms used when you're reading papers about encoding models. Some people call them normalized reverse correlation. Some people just say linear regression, although it's a special flavor of linear regression. Spectrotemporal receptive field is a specific type of encoding model where we're using the spectrogram, which is what I was showing you before. Linear models, forward models, blank temporal receptive fields, STRF, voxel-wise modeling, or multivariate response function. So this is something where you may see any one of these terms depending on the field where you're reading the paper. They all pretty much mean the same thing. There are some flavors of changing how you actually fit the models, but they're all kind of the same. And yeah, for those of you who are saying, thank you for the nice comments on the art. My sister is a wonderful artist, so she actually made some of these, that art on the previous slide. So let's say you've bought into this idea of fitting encoding models. There are a number of different questions that you might be able to answer with this. So here we have, let's say you're interested in how does the brain represent the pitch of a person's, that should say voice. This got cut off. Are phonemes processed separately from pitch in the brain? Which stimulus features does brain area X care about? And then how does attention affect the way the brain responds to speech? So all of these are questions that you could answer with this method. And so this is very nice because there are different ways of kind of using the same natural stimuli to answer many different questions with the same data set. So it's also quite efficient in that way. And there are also a number of people who have not only looked at things like attention, but also how disease processes might affect how the brain responds to different types of sounds. So let's dive into what encoding models are. And then in a little bit, I will show you some results from my own work on what we have found with these models. But just to give you an introduction, and since you're going to be hearing a lot about this in this workshop, let's talk about what an encoding model actually is. So what this does is it describes the relationship between some stimulus. In this case, I'm using a sensory stimulus, an auditory stimulus, but I put this in brackets because it could be kind of a lot of different things. It could actually be other different emotional events, could be visual events, but let's say sensory stimulus and the relationship between that and a brain response. So for the example of the spectrogram, let's say you have an example speech stimulus where this is a sentence, then how does that sentence, how do those frequencies in the sound get transformed into the measurement that you're taking from the brain? And here, this is an example of a time varying brain response. This is just an example of a single electrode from a human intracranial recording. There's something happening over time. And what we really want to know is, does what's happening over time here have any relationship to the sounds that you just heard? Or does it relate to some other aspect of this? So what we can do is we can fit linear multivariate temporal receptive fields. And these are fit using this type of equation. So this is a linear model where we are predicting, for example, some neural data. In this case, I'm going to show you a high gamma response from an intracranial electrode. So this is just a brain signal over time. In this demo, I'm just showing you for an example sentence. So this is the response that you have. And let's say we want to figure out which stimulus features make this response happen. So then we have to know for each time point, what is the stimulus that the person is hearing? And we also have to make a choice about what type of representation we want to use. So for example, in this case, I'm showing a spectrogram. And here you can see the high and low frequencies over time. Remember when we were doing the demo, these look like S sounds. This is actually the sentence, nobody likes snakes. So there's S, and you can see that in the spectrogram. And for this S matrix, in this case, let's say you're trying to figure out how the electrode responds to the stimulus. This F part of the matrix, those would be the different frequency bins of your spectrogram. So you may have, in this case, we have 80 of them. Some people have fewer, but it's as many features as you're interested in sort of encoding independently. And then this part, T minus tau. This is important because we want to know what is the brain response at time T, but we also know that when you listen to sounds, your brain cannot immediately react. There's always some time delay between the sound getting into your brain, going through all of those auditory circuits and being processed. So we actually take this stimulus matrix and we look at what is happening in the past, as well as, let's say, at time 0. And you can choose these temporal offsets to kind of guess how far in the past you would like to look in terms of what you think previously happened that's influencing neural activity right now. So is it 300 milliseconds ago? Is it 200 milliseconds ago? Is it way more than that? And you can fit all of those things at once. And then what you get is you actually solve this equation, and you can do this using a number of different techniques. The one that I use most often is ridge regression, and you get this H matrix. And this is the receptive field itself. And so this is going to have dimensions of features by time delays tau. And in this case, I'm showing you an example of a receptive field that is for spectrotemporal information. And so here we have the frequencies on the y-axis and the time delay on the left. And depending on when you read these papers, sometimes they flip the time axis like I have here. Sometimes you'll see it flipped in the other direction. But the way to read this one that I have here is if there is a large positive weight, then that large positive weight means that energy in the stimulus in the past elicited or causes or drives brain activity at time 0. So the brain activity is, we're looking at time 0 and then we're seeing what was happening in the past that made it more likely that we would see some sort of increase in our neural signal. And then what we do is we use this spectrotemporal receptive field on a separate set of data in order to predict what the EEG or the intracranial EEG would look like for new data. And this is important as well because you could fit a model to all of your data, but that doesn't necessarily mean that that will generalize to if you played a new stimulus and you tried to see, is it always responding to the same low frequency energy in the spectrogram? Maybe it was only for the stimuli that you played. So you want to do this so that you then calculate a prediction on held out data, and you can compute the correlation between your actual neural data and your predicted neural data. And again, this has to be not used in training the model, and you'll learn more about that, I think, in this workshop. And if this looks good, you should have a relatively high R value. If it's around 0, or this prediction kind of looks like junk, then likely that means that you've either not chosen the correct stimulus representation. So maybe it doesn't care about the spectrogram, cares about something else, or you're in an area that is not particularly speech selective, auditory selective in this example. Okay. And when I say relatively as well, I think this is important for people who work with different modalities. I'm not sure how many of you, I think there's probably representation from people who do MEG, EEG, intracranial EEG, maybe some fMRI. I don't know if we have any folks doing spike recordings, but this R value that you expect is actually going to differ depending on the signal to noise ratio of your recording method itself. So this is a pretty high correlation, I would say, overall. You may see them lower for EEG. It doesn't mean that they're necessarily junk, but you have to do some good statistical testing just to check. Okay, so as I said before, one of the things that we need to figure out when we are trying to fit these encoding models is what is it that the brain is caring about? What feature is going to drive a neural response? And so you can make decisions about actually how to take that speech signal, in this case, and parameterize it. So do you want to take the audio waveform and do some kind of wavelet transformation and compute a spectrogram like we saw before? That could work. For EEG, often because the spatial resolution is not as good, temporal resolution is very good, we don't see very strong modulation according to the spectrogram because, being able to record a single neuron with a very tight frequency receptive field is not so common. So there you might want something simpler, like the envelope of the auditory waveform. And that is essentially taking this, and if you were to trace kind of an outline of the loudness curve of this audio waveform, that's essentially kind of what you're doing to get the envelope. You may also want to look at phonemes or phonological features. And that allows us to look at things like, well, maybe this particular brain area cares about the vowel ah, but it doesn't matter what pitch it is. If we use the spectrogram, that would be kind of harder to see because ah and ah would look different in the spectrogram. Versus if you're just labeling which phoneme it is, then you kind of ignore that information about the frequencies. And so each of these levels actually allows you to ask a hypothesis about what information is being represented and how. So let me show you again for this equation that I just had up here. We have the spectrotemporal receptive field right here. And if we were to look at the same electrode in this case, but look at the phoneme or phonological feature receptive field. In this case, my y-axis is a bunch of different categories that represent linguistic properties of the signal. So a sonorant sound so vowels are sonorants, or things where you have this, there's no obstruction in the vocal tract, whereas obstruent would be more like consonant sounds. So here, this particular electrode likes sonorant sounds and syllabic, which is also sort of a descriptor for vowels, but it doesn't respond very strongly at the sentence start. So you can pick any of these features, as I said, and this can tell you different things about where a transformation from more acoustic information to linguistic information might happen. And you can do that by comparing, let's say, the R values for different models, or you can even combine them and see if you add any information by including different features in your model together and stacking them. Okay, so this is the schematic for how you might actually do this. And then this shout out to Chris Holdgraf, who has this excellent paper in Frontiers in Systems Neuroscience, where he describes the difference between encoding and decoding models for cognitive neuroscience. So I think this is a really excellent paper if you still would like to look into how these models have been used. I believe he even has code that goes along with it that you can follow along with. And so here in his example, he's also using ECOG or electrocorticography. So these are invasive human electrophysiology signals. And you perform this feature extraction where you decide what would you like to do in terms of representing your audio signal. And let's say, okay, we've decided to use the spectrogram. And then also, how do you pre-process your neural data? And in this case, maybe you are calculating the high frequency gamma amplitude for ECOG. For EEG, you may be bandpass filtering in the delta band and the theta band and comparing how those track the spectrogram in different ways. So this is sort of up to you. These are different things that you can kind of change. And then when you fit the model, you take some subset of the data, and that's called your training set. And you calculate this, you fit a model where you're finding these weights, W, that describe how the neural activity Y is transformed by whatever your spectrogram, your feature is X. And then B is just some intercept. You may also have some residual noise. And then once you fit the model on your training data, you then evaluate it on a held out set. So maybe it's some sentences that you did not use when you were calculating the W matrix. Maybe it's some podcast stories, or it's 5 minutes, maybe it's some percentage of your data, but it should not overlap. So that's important. And then you get this prediction between your actual Y, which is the real neural data you've recorded for that held out story or held out sentences. And you compare that to your prediction if you were just going to guess based on what the spectrogram was at that time and the weights that you fit. And if it's a good model, as I said, your R value, your correlation will be high. If it's not, then it will look less good. And I'll just take this question right now since it's in the chat. So there's a question where if the stimulus input has to be, I thought the stimulus input had to be a waveform. How would you convert phonemes into a continuous variable? So it turns out you actually don't need the stimulus input to be a waveform, and you don't necessarily, I guess you need a continuous variable, but if this signal, instead of a spectrogram, if it were phonemes, then it would be a binary matrix. So you would have all of your time bins on the x-axis over here. And so let's say you're recording at 100 hertz. So each of these is a 10 millisecond sample bin. And then for that 10 milliseconds, was there an ah sound? Yes or no? And then if there is an ah sound, you put a 1. If there is no ah sound, you put a 0. and you just stack all of your phonemes. So it's essentially a binary matrix where 0 is the feature is not present, and then 1 means the feature is present. And that's why I was saying this is a very powerful method, because it doesn't even have to be phonemes. It could be something else. Yeah, it has to be continuous over time, as Erin is saying, but the values don't have to be continuous in the way that the envelope is. Yeah, thank you. Okay, so let's keep going. So now I want to show you a little bit about just how to interpret these so that when I show you some results, they make a little bit more sense. So this receptive field that we were just looking at before, again, what this is it's a representation of the stimulus features. And it's showing you which of these features in the past drive a response in a particular brain area. So you would have one of these matrices for each area that you're recording from, or your whatever you decide your level of neural recording is at. So you could have 64 EEG channels, and you could fit a separate receptive field for every single one of those 64 channels. You could also average them all together first. If you think that that will give you better SNR, or you have some reason to do that, you could take your average EEG and then look at, what is the receptive field for that. But typically these are sort of one brain area. So you'd have one of these per neural channel that you're recording from. And in this case, this is an intracranial electrode. And so here it is showing that this intracranial electrode really likes frequencies of sound around 0.5 kilohertz and below. And there's maybe even a little bit of structure in there where you see kind of two blobby stripes down there. And so that looks like this may be an area that's selective to vowel sounds, because those do tend to have power in the spectrogram at those frequencies. This particular receptive field shows that this neuron or brain area or electrode would not respond very much if it heard a S sound. So remember when we played with the Google spectrogram and I go, S, S, S. If that happened, there's kind of, the weights are very low magnitude here. They're almost around 0. If anything, it's just background noise. So if you heard a S, this particular electrode is just not going to respond. And so that's the nice thing, is you can find electrodes that respond like this, but then you can also find electrodes that do only like high frequencies and they don't like low frequencies. And if we were doing this with phonemes, you can see some that like fricative phonemes like F and S, or others that like vowel sounds like ah, e, oo. Okay, one more sort of concept that I want to talk about is the idea of speech tracking. And so this is a term that you may hear in this workshop quite a bit. And what this often refers to is a high correlation between a stimulus and a neural response. And it usually reflects the rhythmic tracking of the speech envelope. So this is a different kind of way of plotting the data. This is not showing the receptive field itself. This is showing the correlation, actually a normalized correlation, between the brain activity and an envelope for two different speakers when someone is trying to attend to a male talker or a female talker when they're being played at the same time. And this is a very cool paper showing that one of the very interesting aspects of this method is that it can show you that when you are listening to two people talk at the same time, your brain will likely track the voice that you're trying to attend to. And it will ignore, it will suppress information about that other voice. And in fact, the brain rhythms will just not be correlated with that unattended talker at all, even though it is present. Okay, and this is being used in a lot of cool applications. So again, there's the same framework for many different recording methods. And I'm just going to show these so that you don't come away from this thinking that you can't do this because you're not using EEG. It's not true. I think you can use this for kind of any neural method that you're interested in. And in fact, some of the earliest work on these type of receptive fields came from songbird single units. And so Frederick Tunison and colleagues have done a lot of work on showing responses in bird brains to natural sounds. There's also people who work on ferrets, and they have shown that there can actually be changes in this receptive field based on behavior. If you have to do a target detection task, that can change whether your receptive field has power at a given frequency. There are others, Heather Dial, who is now at University of Houston, was working on looking at how these receptive fields change in primary progressive aphasia. So you can use human EEG, and you can also look at clinical populations. Or some of the things that I'll show you today as well, or you can look at basic tuning to different linguistic and acoustic features in intracranial recordings. The one I don't have here is fMRI, but that is also possible. The only thing is the time scale is much slower. So the way that you parameterize your stimulus might be a little bit different. Okay. And then last part before we really get into the rest of the results is you may also hear about decoding models, or you may have wanted to fit something like this, because this is also a very cool application, but it's kind of the opposite of what we are talking about. And in this case, instead of an encoding model where you are predicting the brain response from the sensory stimulus, you go the other way. And in this case, you would predict the sensory stimulus from the brain activity. And when might you want to use these types of methods? So what I like to think, and maybe others can add to this, is that encoding models really help you to learn how the brain is representing information. So I think this is a kind of neuroscience question. Which areas do what? Which areas care about what level of representation? Are there different areas for phonemes, syllables, semantics, prediction error, all of that stuff? But for decoding, this is more for engineering applications, and some examples of that would be, let's say you want to control a speech assistive device with your brain. So there's been some really interesting work coming out showing that you can take brain signals from invasive recordings in someone with ALS or with a brainstem stroke and convert those into words. So for someone who has lost the ability to talk, but who can still think about what they want to say, this would be an application of decoding, because you take brain activity and you turn it into sound instead of the other way around. There's also some very cool work on cognitive hearing aids, controlling robotic arms, et cetera, et cetera. I'm not going to get into that, but definitely I know some of the organizers have been involved in these types of work too. So there's many different ways to look at this. Okay. So now I want to tell you a little bit about how I have used this in my own work to understand how the brain processes speech sounds. And so I'm going to give you some brief results from a study using intracranial recordings, and then another study using EEG. But it's the same method. They're both using these multivariate temporal receptive fields. They're all using ridge regression. They just use it in different ways. So this, I'm going to talk about this paper that I worked on with Yulia Oganian, Jeffrey Hall, and Eddie Chang that just came out last year. And here we had a number of questions about the difference between encoding in primary and non-primary auditory cortex. So we were able to get some rare recordings where we simultaneously were sampling the temporal plane, which contains human primary auditory cortex, as well as the lateral cortex, which has more kind of classical speech areas. And we also wanted to look at whether functional areas interact during speech perception and whether this supports this hierarchical transformation of acoustic to linguistic information. And so I've talked about this in passing a little bit today, but for all of these, we're using intracranial recordings. And these are typically taken in patients who have what's called intractable epilepsy. And this is epilepsy that cannot be treated by medication. And so these folks come in for a surgery where they have electrodes placed either in or on the surface of their brains to try and localize their seizures. They have these electrodes in the brain for around a week. And then after that time, the clinicians try to find where the seizures are coming from and where they spread. They have the electrodes in for a week though. So we then ask them if during that time they might be willing to participate in research. And so we have these recordings coming from the head, and we can present stimuli on a computer and then measure what is happening in the brain signals over time as they're listening to continuous speech. Okay. And so here is what the setup kind of looks like, or this is where we're recording from. So I want to point out here that we have coverage of the posterior superior temporal gyrus, which has the canonical Wernicke's area, which we know is very important for understanding speech. But then there's also a lot of other areas on the temporal plane that are relatively inaccessible for most intracranial recordings, at least with grids. So in this case, we were able to access this area as well that includes primary auditory cortex over here on Heschl's gyrus. And what we did is we recorded activity from these grids in 9 participants while they listened to sentences from the Timmett acoustic phonetic corpus. And so these are 499 sentences that have that come from speakers across the US. They're also phonetically transcribed. So this is very nice because this data set allows us to look at not only the spectrogram of the sound as they're hearing and as the brain is responding to this, but we could also look at phonological information. And what we're using here to be our brain signal, so if we are looking at the STRF equation and we're trying to model brain responses, then the left side of that equation is going to be this z-scored high gamma. And we have one of these squiggly traces for every single electrode that we record from. And it's going to be much longer than a few seconds. This is going to be over, maybe an hour of data. And this is just to show you what the high gamma signal looks like if you were to decompose the neural signal into its constituent bands. For those of you working with EEG, a lot of the time we work with lower frequency signals that we know also track the features within speech, but it seems like maybe at a slightly slower or sort of smeared timescale. So let me show you a little bit about how the speech representations compare across different areas of the auditory cortex. So what we did in this case is we had, again, these recordings from across the primary and non-primary auditory cortex, and we took the stimuli that people were listening to, and we decomposed them into different feature spaces. So we had the spectrogram, we had just an onset feature, which was the onset of every single sentence. We'd found from previous work that this is actually a very important signal in some areas of the STG. Then we also had phonetic features. So as I described to you before, these are kind of a reduced way of describing phonological content. And then we also had pitch features, including the absolute pitch of a sentence. So that would be if a person has a high-pitched voice, that's going to be a higher number. If they have a low-pitched voice, it's lower. And then what we call relative pitch, which is you take the pitch of someone's voice and you z-score it. So you kind of say, is this pitch higher or lower than that person's average pitch? And you can imagine that there are people whose voices don't overlap at all in the absolute pitch space, but maybe they have a similar range where some people use a lot of dynamic range when they speak, and some people speak in a monotone, so they don't change their pitch at all, and it sounds like this, right? So those two would look different in the relative space. And then we fit a number of receptive fields across all of these electrodes in all of the patients that we recorded from. And here are some examples of what we see and kind of how to interpret them. So this first one, electrode 1, which was here in the back of the posterior superior temporal gyrus, this really just responds at sentence onsets and a little bit to a feature called peak rate, which represents kind of syllable boundaries, but it's not phonologically selective. So here the weights for these phoneme features over the time delays, they look pretty washed out, right? So in this case, it's responding a lot to that onset feature, but this is not very phonologically tuned. On the other hand, we can look at electrodes like E2, which does have a lot more dark purple features in the phoneme feature space of this matrix. And I'm showing a lot of these stacked together because in the paper itself, we actually chose to kind of perform different subsets of combinations of features or just one feature alone. And we tried to see whether if we added those features to our model, if they increased that correlation that we see between our predicted and actual response. And so that's why some of these have more features than others. This particular electrode 4, which is over here in the kind of anterior temporal, not totally anterior, mid to anterior STG. This responds a lot to pitch, both absolute and relative pitch. And then in the phonological features, these kind of dark bands right here are mostly for the vowel features. So that also kind of makes sense because the vowel features have a pitch. So there's a correlation there in terms of which features actually co-occur. And so what we see is that there is this map of acoustic and phonological representations across the auditory cortex. And so we have onset selective areas in the back of the STG. We have more relative pitch selective areas in the sort of anterior and anterolateral areas. Absolute pitch seems to be most common on the temporal plane and not really so much on the rest of the STG. Okay, so this is one of the conclusions of this paper. There were many different aspects to this paper, but essentially what we found is that the Heschel's gyrus, which was the top part here, including primary auditory cortex, shows a really different feature representation than the STG. And we were able to do this using this receptive field analysis. And in fact, those onset-driven responses that I showed you, where it just responds at sentence onset and it's not phonologically tuned, those guys only seem to be in a specific area of the PSTG and not in the Heschel's gyrus. And then finally, that absolute and relative pitch seem to be represented in different areas of the cortex. And so I think, again, this is one of these ways in which we've been able to take the same stimulus and do a lot of different things with it. And so we're able to look not only at acoustics, but we can look at phonological representations, and we can look at pitch representations. Now, I want to leave time for questions. So maybe what I'll do is I'm going to give you the super quick overview of the last part of this, which is a comparison between invasive and non-invasive EEG. And so this is me and my grad student, Mansi Desai, who recently published a paper in the Journal of Neuroscience, where we were using these same methods for EEG studies instead of intracranial EEG. And essentially what she wanted to do was she wanted to compare kind of sounds like timid, where we have a sentence that's in clear, there's no background noise, and compare that to a more noisy naturalistic stimulus. And in this case, we used movie trailers. So they're actually audio-visual, contain a lot of extra stuff. And we wanted to see whether we could use that natural stimulus to look at encoding of acoustic and linguistic features. And essentially, she did the same thing where she took the stimulus, and it's actually the same timid sentences from our previous ECOG work. We decompose them into these phonological feature representations, envelope, and pitch. And then we fit an encoding model where we predict the EEG based on those stimulus features. And when we look at how well that does for the timid sentences, it actually does really quite well. And here, one thing you'll see, these are what's called noise ceiling corrected. So they are very high R values, but they've been divided by the maximum possible R value that you could get. That's why they're so high. And in this case, we do see that you can compare which feature or combination of features drives the brain activity. And in the timid sentences, when we have a model that combines all of the features, phonological features, envelope, and pitch, that's on the x-axis, and each of these dots is the R value for how good this model is. So a higher number means that the prediction fits the actual data better. There's kind of more points that are below the unity line, though at the same time, if we just have the phonological features, which are the red dots, that still does really quite well. So we see this relationship between all of these different features, and we do see nice tracking of that phonological information. So what about for a much noisier stimulus? So we also have these movie trailer stimuli, and we were originally motivated because we have been doing some intracranial recordings with pediatric populations, and these are much more fun to watch and listen to. Kids love them, and so we can actually get a lot more data with these, and they have these other aspects where you could look at differences in tuning for music or speech or different background sounds. So we had to take, in this case, all of the features over time for this movie trailer and then do the same thing. We fit another model for the movie trailers. And we still see phonological feature tracking. These R values are a little bit lower for this stimulus, but they're still quite good. And the phonological features by themselves work almost as well as the full model. So our interpretation for that is that the EEG is able to represent the phonological content of speech quite well, even though you have all of this extra background information going on. And you might think that using such a naturalistic stimulus seems like kind of a nightmare to analyze statistically, but it actually has worked super, super well. And then I think this has also allowed us actually to get a lot more data in our clinical populations. And so that is what I would say is also a huge benefit to using these multivariate temporal receptive field models is you can use those more engaging stimuli, if that's a podcast or a story or a movie, and you can get these same results. And in fact, she even did a comparison between the receptive fields that you get when you use only the movie trailer data on the right or only the TIMIT sentence data on the left. And they're very similar. And this is just a correlation for each of the sensors on the head, for how similar those actually are. So I hope that in this talk, I've shown you a little bit about how powerful these linear encoding models are for understanding how the brain represents our world. And again, as we said, those sensory stimuli, it can be so broad. You can choose, you have to be clever about it, and you could test sort of which features work better, but it's very broad as to what is going to work. And also it can be used on any type of continuously acquired neural data, whether that's from animal single units, human, invasive or non-invasive. And as I said before, this is very powerful for understanding naturalistic stimuli that are so important in our real world. So with that, I would like to thank you, and I'd like to thank the members of my lab. These are the folks who worked on, so Monsi and Jade and Nat and Cassie worked together on the EEG paper. Yulia and Eddie worked with me on the ECOG results that I showed you. And yeah, thank you so much.
Thanks very much, Liberty. That was a brilliant talk. We have a few questions here in the Q&A that I'm going to read out, if you wouldn't mind. Yeah, we still have a bit of time. So somebody asked what variables you would pick to investigate processing of speech registers that differ in terms of acoustic features only, example, pitch, vowel space, etc. Would it be phonemes or spectrogram?
Yes, if you're investigating, so that differ in terms of acoustic features only. So you could, if you're looking at vowel space, I know that I haven't shown this here, but there has been some success in using formant features. So you can use pitch F0, and then you can use F1, F2, or F1 or over F2 ratio. The spectrogram also works very well, but it depends a lot on how high SNR you have in terms of your neural recordings as to which one I would suggest to you. If you're using MEG or EEG, I might use a more simplified space like the formant space or a binned pitch model. You can, this is actually a minor point, but important for people who are thinking about this. For pitch, you could have, so if you calculate the pitch of a sound, then you get, all right, just more pretty pictures. That's not what I meant to show you though. So let me go back to my pitch Where did it go? Okay, so here, if you're looking at the spectrogram is the gray part, and then this kind of cyan color is the pitch of the sound shown as a trace of the fundamental frequency. And that's a continuous signal, except for the times when there's no pitch, you might code that as a 0. I think you have to code it as a 0 rather than a nan if you're doing continuous, because that will usually screw up your model. But a lot of the time, what we actually do is we will not use a one-dimensional pitch vector, but instead use a binned pitch vector, where you take the lowest pitch and the highest pitch, and then you make basically like a histogram of 10 bins, and then for each time point, where in those 10 bins are you? And so those are sort of some ways to do that. But yeah, I would say formant space is good, spectrogram is good. Pitch in those ways that I just spoke about is good. Yeah.
Cool. We have another question from Vishal. In the spectrogram plot of the ECOG neural data, why do we not usually look at visualized stuff below 4 Hertz delta band?
That's a good question. So you're talking about this one. You know, that there's no good reason on this, honestly. I think for us, when we were calculating these spectrogram, so this is, I guess I shouldn't call it a spectrogram because we think of that for sounds, but this is a time frequency decomposition of the neural signal. The 4 cutoff is actually arbitrary. We could have gone lower down to 1 or below, but for the particular pre-processing code I was using, 4 was just the low bound. So that is completely arbitrary.
Great, thanks. Ed Lawler asked, I think I was going to ask this myself, how do you calculate the noise ceiling?
Yes, so there are a number of different ways to calculate noise ceiling. One of them is if you have multiple repeats of the same stimulus, then you can look at basically how correlated is trial 1 of the same sentence with trial 2 of that same sentence later on. And if you had, you know, the highest possible SNR, then they should be exactly the same. Every time you play the sound, you should get exactly the same neural response. But that's not really true. And so you can, I think that's the simplest one, is to calculate just the correlation between each of the possible pairs of trials, and then see what's the average best correlation there. If you don't have repeats, there's some other ways of doing that, but yeah, we can talk more about that offline.
Is there a kind of, sorry, is there a minimum number of trials, repeat trials you'd need to calculate that?
2. I mean, you really want more. You want more than that if you can, but I think people have done it with 2.
I think we've done it with 2 ourselves.
Yeah.
Okay. Thanks for that. I'm going to leave Drew's question, I think, for the discussion session on day 3, because I think it's quite relevant to that. Anna asked, did you use any higher order features as a parameter in your models? For example, word onset. Why not?
Yes, that's a great question. So I think in this slide, I guess we have peak rate is maybe the closest to word onset. It's still acoustically driven, so this is syllable onset. If you use word onset, at least in these areas, it doesn't seem to be the feature that matters most in these areas. But I think that again really depends on where you are recording from. And so actually, if I were recording, well, so this is not as good of a picture for this, but let's say looking at this human brain, I would think if we're looking at more higher order linguistic information or semantic information or word onset, that I think I would see in the more ventral areas. And so those are going to be more sort of language sensitive rather than speech and acoustics. We have done some word onsets in the past, but as I said, they just typically don't work as well, but that's in this area. And so I think that's an important part of the TRF model and kind of a limitation. Sorry, I won't say too much about that, but you know, you have to pick the correct features and you have to kind of think about where you're recording in the brain, what types of neural responses do you think are going to be combined in this area or not. But definitely there's some great work on this using fMRI as well, and EEG, and looking at things like semantic dissimilarity. So I know you guys have worked on that. And so you can get that with EEG, which is recording more broadly. But at least in these areas, I think that for the ECOG recordings, they're a little lower level.
Right. And just before we finish up, I have a quick question. So I was just, you made me think of it there when you were showing that spectrotemporal representation and talking about how with ECOG you typically use the envelope of the gamma band information. And since you've used both EEG and ECOG in your research, other than SNR and being able to be more specific in terms of spatial resolution, what do you think are the main aspects of speech that we're measuring or not measuring with EEG compared to ECOG?
Yeah, from what I've seen with ECOG, it's more difficult to, or sorry, with what I've seen with EEG, it's more difficult in EEG to get strong phonological tuning to very different sets of features. I see phonological tuning in general, but then if I look at the receptive fields themselves, I guess there's some difference, but not nearly at the level of, you know, seeing individual phonemes and some of them being, you know, this is a fricative responsive area or this is a vowel selective. I haven't seen that as much. And the same with the spectrogram. I think it tends to be, they tend to be much more broadly tuned, and we don't see, you know, like those receptive fields that I showed at the very beginning, where it's kind of just very punctate. I don't know how else to describe it, but you know, this is very frequency specific. And so I see less of that. But I think what you gain with EEG is, I think a lot of the, well, first of all, you can do it on anyone. So you don't need to wait for these rare patients to come in and be willing to participate in studies. But you also, I think a lot of the attention signals and things like that are quite strong. And you do have to maybe use a slightly lower dimensional feature space. I mean, I say that, but I know that you and Giovanni have used larger feature spaces. So, but that's my intuition, but I think also it kind of depends how you're doing it. So.
That's great. Thanks very much.
Can I ask you a question, Mick?
Yeah, of course.
Just a quick one. Liberty, thanks a million for the talk. That was brilliant. Regarding data sharing with intracranial data, like I've seen a little, not much in our area, but what do you think about that?
Yeah, you know, it's so tricky because I think the problem is there are a lot of aspects that have to be built into the consent form and they aren't always there. So if they have been there from the beginning, then it's much easier to share. But then I think I mean, there are lots of issues with it. I guess, you know, there's the standardization of formats, which I've also sort of talked to people about and we're trying to be better about, but it's kind of a little bit the Wild West in terms of how people store their data, which systems they get it from. And so that's an issue. And then the sort of privacy related issues, making sure that it's sufficiently anonymized. And I think for me, I would say sharing data that is mostly listening to sounds should be overall easier in terms of privacy concerns than, for example, we also have some data where people are talking and we measure the responses while they're actively saying something, and then we compare that to when they're listening, but then that has their voice. And that's the most useful way to share the data, would be to give you their actual waveform. We could give you the transcript, but that only gives you so much. And so I think that's why a lot of the labs are more like upon request or with a data sharing agreement, I think because of these privacy concerns. So it's, yeah, I think we're trying to be better about it, but I also understand all of the concerns of privacy and potentially actually making it difficult to de-identify some of the data.
That's great. Thanks a million for that, Liberty. That was a great talk, and thanks for answering all those questions for us.
Sure.
Yeah, it was great to have you.
Yeah, thank you so much. All right, look forward to interacting with the rest of you. Enjoy the rest of the workshop. Thanks again for the invite.
That's all. Thanks. So now we are going to hand over to Joe. He is going to go through environment setup for the tutorials and talk a little bit about all the resources up on the CNSP website.
Yeah, so here I am. So thank you again, Liberty. That was brilliant, at the right pace, comprehensive. I love that. So I'll give a presentation now that is about a number of things. Sure, there is the component that is more practical that I'm going to explain, but I wanted to give you also some background on what we're doing in terms of the broader CNSP initiative. So why are we, what's the point of doing this thing? And what motivated us to do this? What is our approach and our objectives? So first of all, a practical side of the story. So if you could please download the resources that are highlighted here. So this is on the resource page of our website. You have done this, or you should have done this already in theory, but maybe some of you had, a couple of you are joining the tutorials a little bit late. So I'll just say this again. I'll plot this at the end of the presentation as well. But one thing to know is that we have updated, and there are a couple of small updates to the libraries. So if you could, even if you downloaded this already, just download it again. And also we have added, if you see here on the 3 stars next to the data set, natural speech, you have an updated data stim with a number of new features that are going to be used in the intermediate tutorial. Also here you have the guidelines still on the resource page of the website. There are 2 documents that are relevant to the workshop. One is the resource preparation guidelines, and the other are guidelines for converting your own data set to our standardized format so that you can run your scripts. And I'll talk about that in a minute. So Liberty brilliantly described this idea that, when we run these experiments on natural sensory perception, for example, we have on the one hand the neural signal, and on the other hand we have the continuous sound, which could be speech, music, it doesn't have to be sound, as Liberty said, it could be video, it could be lots of different things. I will stick to sound just because that's what we do, and to say one thing to keep things easy. And then we have some pre-processing, which could be EEG, MEG pre-processing of different kind, which is similar. It can be similar to what we do already for ERPs, for example. But then we have the additional thing that, differently from, in ERPs, you have your events, and you just need to know where the event is, how long the epoch is, and what condition that event corresponds to, and then you average them. And those are the main things. See, here we do a proper feature extraction, a little bit more similar to what we would do in speech recognition software, for example, where we actually take the speech or the sound or music. We can extract some features and we run these models that Liberty talked about. And so there are encoding models. And just to give you my personal perspective, for who doesn't know, what I have been mostly using is what Ed Lawler, who is in the call actually, and Nick have been sharing, so the MTRX toolbox. And the main point there is that Liberty showed a lot of temporal response functions, or spectrotemporal receptive fields. We can call them in different ways. As I said in the chat, spectrotemporal receptive fields is a fairly specific term that is specific to our field, while temporal response function is a broader terminology that can apply to lots of different things. So in this case, they mean the same thing, really. I tend to use receptive fields for more intracranial things. But the concept is the same. So Liberty showed a lot of these response functions, like in this case, the envelope temporal response function. But the point is that to study these weights, the first thing that we always need to do is to make sure that we are actually predicting some EEG data. So if we have a good EEG prediction or MEG prediction, then we can study the models and the coding models that Liberty explained. There are other types of models that we will cover in the intermediate tutorial. For example, the canonical correlation analysis models, where instead of having just one transformation, so in the forward model, you transform the speech to predict the EEG, and in the backward model, you transform the EEG to reconstruct some speech features. Here, you're transforming both in a way that you transform speech in a way that you preserve only what is relevant to the neural signal, and vice versa. You also, for the neural signal, you just keep whatever is relevant to the speech, and you calculate the correlations in this canonical correlation space. And this is all work that I learned from one of my mentors, Alain de Sévigné, during my postdoc in France. So we are getting to the end of session 1, and my point is also to prepare you for session 2. So basically in session 2, there are going to be 2 tracks. There is going to be a beginner track, and there is going to be an intermediate track. So the beginner track is going to cover forward TRFs, backward TRFs, and depending on time, also simple multivariate temporal response functions. And Mick will guide that track. And then we'll have the intermediate track, where we will talk about tuning multivariate temporal response functions, and so banded regression, for example. Also the evaluation of the temporal response function, which is a very important topic. How do we actually quantify how good these temporal response functions are? There is not one way, there are many ways. And as Liberty said, the correlation values we get with different neural signals change a lot. How do we know what is meaningful and what is not? There are techniques to do that. And finally, CCA. So the rest of the presentation is about talking about a standardized framework that we are proposing for the workshop and beyond the workshop. Then I'll talk about the CNSP resources. And finally, I'll give a slide on the call for projects, which is supported by mBrain Train, who's going to give us some funding for a travel grant for the winner. So let me start with what happens when a person joins our lab or your lab. Usually this person joins the team, runs a new experiment, or reanalyzes an existing data set. And then maybe the supervisor gives them some lab-specific core scripts, so some scripts that the supervisor or the team uses. And the student is going to make this theirs, and they're going to develop some custom code that is specific maybe to their experiment. So sometimes the student is actually going to contribute to the core scripts of the lab, and so improve the actual set of scripts. And many times, anyway, the student graduates before any of that happens. And so really, with the next student is going to be the same pipeline, and we don't get much improvement. So this is just within lab, but you'll see how it's relevant in general. So one key aspect is sharing. So it's in our field, it's relatively recent. It's a relatively recent effort, this idea of sharing data set and toolboxes. I mean, to be fair, we have been doing it for years, but specifically this area of natural sensory perception is relatively new. And so only now we start having a good amount of effort in this area. Anyway, the main point here is within a lab or between labs, we may have many data sets and each data set with their own particular custom data structure, maybe very similar, but these small differences lead to also more different analysis code. And so we have all these codes that are incompatible, so you can't take one and run it on the other. And so then what happens, And some of the PIs in the call that know that they get a new student, and it's easier to tell the student to start all over again and convincing them to try to do it better. But sometimes reusing code is just hard, mostly because PIs don't have time to rewrite the code of their students. So the ideal situation here would be that, if you have many data sets, again, within a lab or ideally between labs, and then you have some standardized analysis code for one particular application that we may be caring about. And this analysis code, the code is always the same, but we just change some parameters, and this code then applies to all of these different data sets. We're not trying to reinvent the wheel, but by changing little things, we can actually make things much better and faster. So it's a difficult challenge, though, to find agreement on how to do things, but the benefits are potentially great. So one thing to understand, and this is not about just what we are about to talk about, but it is a general concept of standardization. So this applies to a number of fields. And there is this trade-off between how flexible your standardization is and how simple it is to use. So you may want your standardization to apply to a number of different types of data. So maybe EEG, MEG, intracranial EEG, and different tasks. And that's beautiful, right? A standardization that applies to a lot of different situations. The problem there is that the standardization maybe requires a lot of options, lots of parameters. It becomes really flexible, but also maybe a bit complicated. You need to read lots of tutorials, and it just turns out that maybe two people that are trying to do the same thing, they may be doing it in a different way within the same standardization because the standardization is so flexible. And this is not just me waffling, this is actually what you can see in some of the data sets that are available. So one point of standardization in general is to facilitate replication. But the other point is also to have faster analysis, reusable code, more robust code, robust to programming errors, building graphical interfaces like EEG Lab did. That's all pretty standard objective, let's say. But what is new in what we are trying to do with the CNSP initiative here? So the new thing, as Liberty said, one thing that Liberty said that is striking is that it's a bit of a Wild West here, right? In terms of standardization, there is no standardization in what we are doing in terms of natural sensory processing experiments, and in many cases also in terms of event-related potentials. Many people are doing their own thing, and there are some standards, but every experiment is just so specific that usually what you do with the purpose of sharing some data, like on MMN experiments or on other event-related potentials, usually is to replicate the study. And so to check that everything is all right, maybe with a different type of analysis, but still constrained to that particular task, that particular investigation. While the unprecedented opportunity here is that we can actually reanalyze this data based on natural speech listening, for example. We can reanalyze it in 100 different ways, and because there are many simultaneous neural processes going on in our brain as we listen to speech, and the single lab is unlikely to have time, expertise, and interest in analyzing all of these processes. Personally, I'm interested in a number of things, from acoustics to phonetics to word surprise, melodic expectations, and so music listening. So a number of things, but it's still fairly small compared to the number of things that one could study. There are prosodic aspects, emotional aspects, tension, and all of that. When we talk about movies, like Liberty talked about earlier on, there is even more that could be studied. So it's just a shame that one would collect data set to just study one aspect, and then that's it. The data set gets lost or gets just So I see that Liberty is replying, the reanalysis angle is great. The number of papers with timid ECOG dataset is quite something, yeah. So data sharing has great potential beyond replication, but sharing, as Liberty said earlier on, has to be done right because it's quite the Wild West. So that's the problem we're trying to solve here. So typical issues with currently available data sets: no documentation. Then an even worse problem is poor documentation, because in addition to not having documentation, you're wasting your time trying to understand very badly written documentation. I'm criticizing myself here in some cases. Poorly written code that does not run, with absolute paths, weird naming. And then using custom data structures, so no documentation on the data structure. And finally, one important thing, oftentimes we share the pre-processed data. This is a discussion we had at the workshop called COG here. And the discussion was also led by, especially led by Alain de Chevenier. And in that case, if you share the pre-processed data, the main thing you can do is to replicate the result that was published. So sharing pre-processed data is great, but it's much better to share the raw data with the pre-processing code. Because if you want to reanalyze, you may want to reanalyze with different parameters. So, you know, it's a big ask to share data sets that are, you know, hundreds of gigabytes. So maybe we can apply some mild pre-processing, some basic low-pass filtering and downsampling, just for bringing this to a tractable level of, you know, how much data we're actually sharing. But generally, we should be sharing raw data. So the result of all these problems is that the datasets can't be used, and people get in touch with the authors multiple times with multiple interactions to try to get them work. And I'm talking about myself here, because in the past I shared datasets in ways that were okay, but not super. And so it was impossible to use them fully without getting in touch with me. And so I'm trying to improve that aspect, of course. Now, a pretty picture that summarizes a bit. This is on the left is the lab-specific approach, where we do have data acquisition. We go onto the raw data, which is specific to the device. So that's layer one, device specific. And then we typically import this raw data with a custom importing script that goes straight to a domain specific. So it's an import script that takes whatever we care for our experiment, and then we run our custom analysis script and we get our outcome. So of course this works, but it's not really reusable if not well done. And it can work within a single lab maybe. The standardized approach. So this is not stuff that I did. This is the field, right? There are data structures such as BIDS, for example. So BIDS is great, mostly used for MRI, but also recently used for some EEG and a little bit of intracranial data, which turned out to be not my favorite data set, in the sense that I couldn't use it for anything because it was very little data and there were not the original audio files. Anyway, so you can import your raw data onto these standardized data structures, which I call structures at layer 2, so that are device and domain independent. So they are independent from the device, but also independent from the particular application. Are we talking about continuous speech, or are we talking about ERPs, or we're talking about some other analysis with fMRI? So BIDS can capture a number of things. So then we need to go through a domain independent processing to extract whatever we need, and then we run some custom analysis scripts. And so we are back to square one. Because these data structures are great, but they require some specific domain-specific pipeline afterwards. And so that's what we're trying to propose here, right? Can we make this domain-specific by either taking BIDS and importing it to whatever we need for our analysis, or in some cases we can jump directly to whatever we need straight away without going through BIDS. Now, bits is great, especially if we run experiments that have multiple things. Imagine an experiment where we have, EEG, MRI, source localization, or MEG, MRI source localization, and maybe eye tracking and those things, and other sources, of information. Then bits is great because it allows you to, it's flexible enough to capture all of that. And so then we can get the domain-specific import script that we are working on at the moment. We have a version, but we haven't shared it yet, that transform this onto a standardized data format that is specific to the CNSP area, let's say. So the continuous, the natural sensory processing experiments. And then we can use our CNSP libraries that we are proposing here, that we are giving you as part of the tutorials, to analyze these data structures. So this is not complicated. I mean, it's much more complicated to explain than to actually run this. So you'll see in a few minutes. So we are starting with MATLAB because that's what we use, what I use at the moment, but the same is valid across other languages. Python, and Christian Brodbeck last year helped us to integrate this same pipeline in Python as well with his EELbrain toolbox, which is going to be shown tomorrow by Joshua. So very simple. First thing, what are the folders? Oftentimes students and my students and people tend to underevaluate the importance of having a proper folder structure. So it's important. Use the exact same names all the time. And for example, here is tutorials, datasets, libraries. That's it. Very simple. And within the dataset folder, so within the tutorial folder, you have, for example, a tutorial script. So if you download the skeleton of the CNSP tutorials, you will find this. They are empty folders, but you find these folders. And then you can download the data. So datasets in this standardization that we're proposing, and that we used last year as well in the workshop, datasets are organized like this. You have a dataset folder like Dilibach or Lawler natural speech, and inside that folder you'll have a folder called data CND. CND stands for continuous event neural data structure. So continuous event because it's not a discrete event like an ERP, but it's a continuous event like speech or music. It doesn't have necessarily to be sound, it could be other things, but I'll stick to sound for now. So the only thing we need in here is this folder, data CND, with these standardized files. You can also have, and this is optional, your original stimulus files and your EEG, raw EEG files, for example, that were directly recorded from the Biosemi or brain train device that you're using, or Brain Vision. So the data CND is still raw data. It's just chunked onto different trials. So when you look at data sub1.mat, you have data, it's the EEG data or MEG data for subject 1. It could be intracranial data and so on. So you have a structure inside this file that contains mainly 3 things. One is the sampling rate, which you always should know. So nothing new from what you already do. Then is the channel location file, which tells you how many electrodes, how many EEG channels or MEG channels, and where they are. And then the most important thing is the data. So the data is just the raw data that is chunked into trials, where trials can be, for example, chapters of your audiobook, if it was an audiobook, or chapters of your movie, or experimental trials in this case. So N trials with capital N, and then you have that each cell, so each trial is composed of time samples by channels, exactly like you would do for any other EEG experiment, really, with epoching. And then you have your stimulus, where, again, same thing, you will have the sampling rate, you have your data, and then you have your names. Now, in the stimulus, it's a bit like Liberty was showing earlier on, right? You may have multiple stimulus features. You may have, for example, the envelope, the word onsets, the phonemes, phonetic features, word surprise, melodic surprise, pitch, lots of different things. So each of those features can be a different element of these M elements. So the name says what that element, what that feature set is. And then each feature set, there will be, of course, for every trial of the N trials, you'll have time samples and then feature dimension. Where feature dimension is, if you have envelope, feature dimension is 1. If you have spectrogram, you'll have maybe 8 bands or 16 bands. And so it can be multivariate. You can have a combined model like Liberty was showing. And so that would have a combination of all features here. And it will be clear when you run it. You can also find the CND specifications on our website on the resource page. Yeah, so if you go on the resource page of the CNSP workshop, you'll find this table. Now it's still old looking in terms of style, but all the information is there, and we'll try to do our best. We just didn't have time to do this fancy. But the main point is you have your tutorials for this year, and then you have your data sets. So that's everything you need for the workshop, really, these 2 sections. Then you have down here, you have the toolboxes, the original links and the papers. We're trying to be fair, not to skip anything. Now we have been working, so with the help of 2 students in my team, Sarah and Amir, we have been working on the conversion of data sets from other teams onto the CND data structure. So on this table, you'll find on the left-hand side, the original link to the data set, the paper in the middle, and on the right-hand side, you'll find the link to the CND data structure that you'll need to download and then add to the dataset folder if you want to analyze with our scripts, or with whatever scripts you work on that are compatible. So Sarah converted this dataset from Alexander Bertrand on auditory attention. We're working on other datasets, another dataset from Ed Lawler, and then Amir is working instead on a conversion, on a more standard conversion from bits to CND. So we have a version. We haven't shared it yet because we need to double check things. I'm almost done. So, of course, more data sets will be added in the future. So the worst case scenario is that, the data sets from my own team and from the CNSP organizers will be added in the future to this table. So, in a few years, we'll get, 30, 40 data sets through the help of the CNSP organizers. The better case scenario, because parenthesis, I'm using all of this standardization within my team, and I find it extremely useful. So I'll just keep using it. So, but the better case scenario is getting your help, and we do need your help to add your dataset to the list. If you have datasets that are already shared and could be converted into CND, or you may help converting other existing datasets to CND and help us enriching this resource, or adding your CND compatible analysis code. So all of that could be of great help. So why is this useful? And one thing that there are just a couple of things that I'd like to mention. For example, imagine having 2 different experiments, so 2 different data sets. Well, you can run these data sets CND in CND format through the same pipeline, same preprocessing, same analysis, TRF, CCA, whatever you want that is available, and you get an outcome for each of these data sets, and then you can compare them. So it is key to define an objective and fair comparison metric. And so there has been a beautiful discussion about this led by Alain de Chevenier at the COG here workshop last month or 2 months ago. I lost track of time, where we were in fact discussing what these metrics could be for comparing different analysis, different data sets. So we'll discuss this a little bit in the intermediate tutorials. But here you have a couple of figures that you can get from the example tutorial script that we have on the skeleton of the CNSP workshop. So You have on the left the forward TRF for the natural speech data set. This is a data set where participants were listening to The Old Man and the Sea. So I recorded part of that, and the other part was recorded by Michael Broderick. So it was published a couple of years ago. So it's out on 19 subjects, and you get these beautiful temporal response functions. I won't explain the details now, but it's more or less what Liberty was showing. In this case, on the top, you have the prediction correlations for different participants. So you see that, as Aaron mentioned, and Liberty mentioned, the prediction correlations are, 10 times smaller than the numbers that Liberty was showing. Now, to be fair, Liberty was showing a particularly good electrode. I work with ECOG, and yeah, they are higher, but maybe not 0.6 all the time. But yeah, they are much higher than this. So first of all, you run these, you can get the prediction correlations. We'll see in the intermediate tutorials how to assess whether this is significant in detail, especially with multivariate models. And then you get these temporal response functions on the left for this natural speech listening task, and on the right for this time reverse listening task, where speech was time reversed. So you can compare the two. You can compare two different data sets that use different materials. So this is The Old Man and the Sea on the left, Alice in Wonderland on the right, different experimenters, different recording facilities, different device. And then you can also run compatible code. There is a different script for this, but very similar, for auditory attention decoding, where in this case, people were attending to the speech that you see on the left here, and ignoring the speech where you see the TRF on the right here. So you can do all of this with the tool with these tutorials. The other perspective, and Nick, I'll be done just in a minute. I see that you're there. And so the other thing we can do is to compare methodologies. And so we won't do it in this particular edition of the workshop, but the idea is, yes, you have a CND data set. You can pre-process it with a script that is the same for whatever toolbox we use afterwards. And then we can analyze the data with MATLAB, MTRF toolbox, with the EELbrain toolkit from Python, and then compare the methodologies. And one thing that we're working on to do that, because it's difficult, we can't ask everyone to install all different toolboxes on their machine. So we are working on a graphical, a web application that allows us to do that. So that is interfacing with MATLAB. And so people that don't have MATLAB license can use this. And same thing for Python. And then we can compare them within this space. So why is this useful? Why is this going to be useful? Because for the PIs that are in the room that are writing grants, or for the postdocs that are in the room that are writing grants, if you want to write a grant and you want to do power analysis, well, at the moment, we kind of do power analysis on previous data sets that are existing. However, sometimes there is no previous data set or previous papers that use that exact configuration that we want. So how many subjects do we need to get a significant effect? So this is practically a way to simulate with existing data sets different configurations on different data sets, and run power analysis, for example, or get some preliminary results on an experiment that you want to run, and you may want to know how many subjects, how long do the trials have to be, and so on. So all of this is going to be available at some point, and we're working on it. Now, before I show you the last two slides, let me just, I don't want to forget, I just want to thank all the people that helped us. So the speakers from last year and from this year, of course, and all the others that helped us with testing the code and also with converting data sets and so on. Last two slides. One is a reminder about Slack. So you can use Slack for interacting. That's what Nick explained before. It's great for the workshop. After the workshop, we are not going to check Slack, or better, we can check it every now and then. But after the workshop, the easiest thing would be either to email us. But if you email me, maybe I'm away, I can't, and reply in that moment, and you get a slow reply. While here, more people will monitor it, and the idea is that we'll be able to give you a reply that will also be searchable through Google, for example. So we can build a sort of Q&A resource there. So we're trying to launch this. I don't know if this is going to work out. We'll try it. And so if you have questions after the workshop, you can try that. The other thing is, this is about the CNSP workshop. It's not about, so don't ask questions about technical details on the NTRF toolbox. We have the GitHub on the NTRF toolbox for that, same thing for the other toolboxes. So this is more about the general questions on the CNSP scripts, on the theory, and so on. And finally, the call for projects. Now we have a call for research proposals open that will close at the end of the month. Now, the idea of the call is that mBrain Train is sponsoring this. And they are giving us about €1,500 to support a travel grant that ideally would be for a student that proposes a project to be carried out here with me in Trinity College. And it is also possible to submit an abstract for a project with someone else, as long as you have the approval of the advisor. So this is going to be, and as long as it's going to be supportive of the CNSP initiative. So if you collect data, the data is going to be shared on the CNSP website and so on. The call closes at the end of July, and it's just a short abstract about about the project that you're working on, that you would need help to work on, or a new project, a new idea that you would like to do in the area that we are talking about here. So speech perception or music perception or something related. And yeah, I'll just leave this slide up so that you can remember what to download. And just a reminder, re-download the libraries because there have been a couple of updates. And I thank you all for the attention in this first session. I'll give it back to Mick. And yeah, I'll talk to you in a bit. It's all yours, Mick.
Great, thanks for that, Joe. So that concludes session 1.